Good morning. We're going to kind of review a little bit about DNA, RNA, and protein synthesis. I know we had this a little bit last quarter, last semester, but we're gonna kind of review it just a little bit, just so you have an idea. And I'll include some extra information here towards the end. So, a little bit about the importance of the nucleus, since that's where your DNA is housed. A gentleman by the name of Joachim Hammerling, he looked at this species of algae called Acetabularius, and he did a couple of different experiments with them. First, he grew the algae, and then he cut it in half, and each gave rise to a new algae, but only if he would cut it parallel or straight up and down, not he had to cut it vertically, not horizontally. So because if he did cut it horizontally, the organism that would grow would only grow. You'd only get a new organism from the bottom half, not from the top half. But if you cut it, like I said, vertically, then he ended up with two separate identical species there. So then he decided to take it a little bit further, and he worked with two particular species of Acetabularius, but he then would cut, he would go ahead and try to cut and grow the algae, but what he did is he took the DNA from one species, because he noticed that the newly grown version was basically identical to the parent. So what he did is he took the DNA information, because he was able to figure out that all the hereditary information was stored in the nucleus, and he transposed that into the second species, and that second species ended up developing into a hybrid of both species, because the DNA was mixed together in that instance. So he was kind of the first guy to really determine that the DNA was located in some sort of structure, which we now know is the nucleus. But to kind of go through a little bit of review here, your nucleus is the structure that contains our hereditary information, and that's what is passed from generation to generation, from parent to offspring, whether it's through asexual reproduction and the parent becomes the offspring by dividing, or it actually gives rise to and creates an offspring itself there. So, and then your nucleus is also responsible with that hereditary information. It's also responsible for regenerating any tissue that you have that needs to be either repaired or grown more of. So it will also contain information about the organism's growth and form, because obviously when you're very young, you do not look like what you're going to end up looking like when you're an adult, in the adult form. So your nucleus, specifically your DNA, is going to have that information. One important thing to note is that your DNA never leaves the nucleus. It contains this hereditary information, but it never ever leaves the nucleus. So we've got to have a way from that information to get from the nucleus, from the DNA, and somehow make a copy of it to get it out into the cytoplasm, where you're going to build proteins for the cell. And that's where protein synthesis comes into play. So we talked about this last semester, but protein synthesis is just the process where DNA is going to copy its information onto a piece of RNA, and then the RNA will then translate that from the genetic code, the genetic language of bases, of nitrogen bases, into the language of proteins, which would be amino acids. So the very first step is to kind of remind ourselves of the difference between DNA and RNA, which I have on your diagrams here. I've just got kind of a list there that DNA is double helixed, RNA is single-stranded. Sometimes it forms a helix, sometimes it doesn't. Then you've got your nucleotides. They're essentially the same thing, sugar base and phosphate. If you look at on the back side, no, it's the second page, just kidding. So if you flip over, you can see that I've given you kind of the organic chemistry structure of your DNA and RNA sugar specifically. So that's one of the main things that separates or differentiates DNA from RNA is the sugar itself. So in DNA, you've got deoxyribose, which is there on the left column or the right column. And then on the right column, you have RNA or the ribose sugar. So the only difference between those two sugars, as you can see, is the difference of one oxygen there. So that's the only difference. Deoxy means without oxygen. Ribose implies the sugar. So their sugars are just slightly structurally different. And the phosphate group is going to be the same. You can see that attached to the ribose and the deoxyribose kind of off to the left. It's just that phosphate, a phosphorus molecule with 4 oxygens to make a phosphate group. And then your nitrogen base, which is attached kind of above. So your bases are, for the most part, identical, if you recall, but there's one base that is different. In RNA, you use the base uracil, but in DNA, you use the base thymine. So adenine will pair with thymine in DNA and uracil in RNA. It's a double bond, which I've noted there on your sheet, and then cytosine and guanine are going to form a triple bond, which I think we talked about, though. So, but that's your nucleotide. So that's what DNA and RNA are. They're nucleic acids, which are built from these nucleotides. So, kind of a crash course there. So what's going to happen in this first part is your DNA is going to copy its information onto RNA. That RNA then will leave the nucleus through nuclear pores to go to the cytoplasm to make the proteins, which will occur out in the cytoplasm. So there are 3 different types of RNA. I know we've talked about those: messenger RNA, transfer RNA, and ribosomal RNA. All of that information is coded for on the DNA. So the DNA strand will make all 3 of these types of RNA. The messenger RNA is really the template or the actual copy of the DNA. So there are 2 different types of proteins that your cells will make. It's either going to be a structural protein or it's going to be an enzymatic protein. If it's a structural protein, we're looking at things like hemoglobin, collagen, keratin, things like that. An enzymatic, self-explanatory, it's going to be something like cellulase or amylase, a protein that's going to serve as an enzyme. It's going to break down something or promote a chemical reaction of sorts. But a structural protein is just there. So therefore the structure of your cell, like collagen. So okay, so your next set here is the actual process of protein synthesis. So we're going to start with transcription. The process is divided into 2 parts: transcription and translation, which I know we've talked about before. But your transcription is the first part, and I've drawn that up above, or sorry, not up above, but in the middle block there of your diagrams. You've got your DNA here. So this is the process where DNA is going to replicate itself onto messenger RNA. So you can see I've drawn 3 little arrows. Your DNA will code for the messenger RNA. It will also create the transfer RNA that will be necessary in translation, and it will also make the ribosomal RNA, which will be necessary also in translation. Now what I've drawn you here is, remember, the ribosomal RNA is constructed here, but it's split into a large subunit and a small subunit, because if we put them together in the nucleus, it's going to be too big, and we won't actually be able to fit out of the nuclear pores. So that's why they're not connected there. And if you recall, they're made in your nucleolus. So again, important to note, DNA never leaves the nucleus. So all right, so that's basically transcription in a nutshell. We're not going too far in depth since we've already gone over that before. So in the previous semester. The right side of the diagram there in the cytoplasm, this is going to be your translation part. So what's going to happen here is you've got your messenger RNA, which contains the codons. If you remember, the codon is that series of 3 bases. I know I told you that in the past your DNA, or your ribosome can only read 1 codon at a time. It actually can read 2. It's actually only reading 1, but it's anticipating the second to start lining up that transfer RNA. That transfer RNA is going to have that anticodon, which is going to match up with your codon on the messenger RNA. So, and then its anticodon is very specific as to what amino acid it's going to pick up. The amino acids are going to come from the food that you have consumed. Your enzymes will break those apart. So your steak that you ate, your enzymes in your body are going to break those down into individualized amino acids. Those are then going to go into the cytoplasm and kind of just hang out, waiting for that transfer RNA to come pick up that particular amino acid. So this is not the best example in the world, but we're familiar with that you have tryptophan in your turkey. So when your body breaks that down, you've got the tryptophan that goes into your bloodstream, which goes into your cells, and then that tryptophan is going to wait and hang out until it finds the transfer RNA that's going to pick it up, and then that will start making those structural or enzymatic proteins that your body needs. So that's why protein is important for your body. So that way we can make more keratin and collagen, and we can make more enzymes to do the various functions in our bodies. So that's actually what happens. Your ribosomal RNA, before translation technically starts, if you recall, because you've got to have it to be able to do translation, you're going to assemble those subunits together. So a ribosome is just ribosomal RNA and protein. So there are some little small proteins embedded in your ribosomal RNA to make up your ribosome. It's not just ribosomal RNA. So, like I said, I know that was kind of fast, but I know you've had this before, so I'm not going to spend too much time on it. Okay, so I'm gonna fly through this one too here, but there's 3 main parts in translation, which is initiation, then you go into chain elongation, finally you have chain termination. Okay, so with your initiation, this is how your protein starts being built. Your start codon is read. Methionine, the amino acid, is brought in. That's initiation. That's it. Chain elongation is just going to be the rest of the ribosome is reading the rest of that messenger RNA strand and translating, because you're elongating or building a chain of amino acids. And then finally you have chain termination, which is where the ribosome reads that last codon and stops reading, and then your protein's built, snips off, and then you've got hopefully your fully formed protein. So translation is composed of initiation, chain elongation, chain termination. If you look at the last little parts on the front page that I've got for you on your diagrams, I've got just kind of structurally, it shows you that your protein is just that long chain of amino acids. That's what those AAs stand for. And then the bottom part is just another way for you to kind of look at transcription and translation. So I won't bore you by reading it, but it's just another way to kind of see how transcription and translation occurs. Okay. And that's, if you look at the back of that second page, I've copied that whole bit for initiation and chain elongation and chain termination there and that. So it's probably a little more detailed than you really need, but you know, we'll go ahead and include it anyway. Okay, last little bit for you for today. Transposons. This is the new part that you haven't had yet, as far as I know. Transposons are little pieces of DNA that have the ability to insert themselves into another piece of chromosome. They can take themselves out of one and jump, quote unquote, into another. So we call them jumping genes. They were discovered in around the 1940s by Dr. Barbara McClintock. She was looking at corn, specifically Indian corn. And so the reason why this is so interesting, why this is so remarkable, is because they can insert themselves, they can increase or decrease their ability to be expressed. A gene, I guess I should say. Either themselves or a gene. So, if you look now, I know I'm flipping back and forth. If you look to the back of the hand-drawn doodles, I've got a picture that kind of explains the Indian corn. The Indian corn is a prime example of the transposons. It's the easiest example. So Indian corn has several different colors: purple, yellow, white kernels. And then some of the kernels actually look mottled or streaked, mottled meaning netted, or they kind of look streaky between purple and yellow or purple and white. Let me see if I can, we'll see if this screen shares. Here, let's do. See if we can see this. Okay, so let me see. Okay, good. You're seeing what I'm seeing. All right. So this is what I was kind of talking about. You can see the white, I think this is white, and the yellow corn kernel, and I don't see any purple, pure purple corn kernels. You can see how this kind of looks streaky here and up here. Okay, so these streaky kernels are the result of a transposon. So what happens is normally your gene is going to be transcribed and translated, and you're going to end up with a plain colored kernel like this. Or if I had a purple, all purple one, which they're not showing this one, but if there was a plain purple kernel, that would be another example, or right here. And so you get the full color. Every cell is producing that particular pigment. Okay. With a transposon, if you look now back at the hand-drawn versions that I have for you, on the right side, the DNA with the transposon or the mutated gene, that transposon has inserted itself in between that gene information there. And so that when the ribosome is translating and transcribing, it can't code consistently for that purple pigment, so you end up with a white kernel, so you don't get any pigment produced whatsoever. Okay, so the result of having some genes going ahead to producing that purple kernel, and some of those cells and genes, so some of the genes in those cells not producing the purple pigment, you end up with a white kernel. And so this is where you get that. But then this shows you how the genes can end up coding in one kernel. So in this case, every single cell had that transposon. Here, not every kernel had the transposon. So this area right here produced the purple pigment, normal genes. These pigments right here, or these cells right here, have the transposed genes. So they aren't able to produce that purple pigment. So you end up with that netted or streaky appearance. So that's what transposons are. Sorry, I misspoke earlier. So the normal gene will always produce the purple pigment. It won't produce the white or the yellow kernel. That's going to be a result of the transposed genes. So my apologies there. And sometimes, and this I'm not showing it, but not only can they appear streaky, but they can appear speckled or spotted. So in this case, it would be more streaky, I would call it. So that kind of races you through and reviews DNA, RNA, and protein synthesis. So if you have any questions, don't hesitate to ask. Have a good day.